Hello everyone, have a nice day. Today we're going to be continuing our discussion on the Bio 134 or the biotechnology. And as a sort of review, last time we discussed about the different tools used in biotechnology, particularly in the recombinant DNA, the tools on recombinant DNA. So as a sort of review, let us have a quick review and a quick look on the previous discussions. So, in previous meeting, we have discussed the different tools of biotechnology for recombinant DNA. See, in this particular example, we have in here the sorry, let me have my first my pen, no? Because I wasn't able to emphasize my points without my pen. So I have to insert my pen first. Yeah, there you have it. Okay. So yes, we have the different tools used in recombinant DNA technology in the in biotechnology. So let me have my pin. All right. Okay. So like for example, we have the tools for restriction enzyme. We have the polymerase enzyme, the ligase, vectors, and host. So restriction enzyme used to cut, the portions of the DNA. Used to cut certain genes, in the DNA. And polymerase enzymes is used to extend what's we call this the extend the strand, the DNA strand, that has been cut by the recombinant restriction enzyme. The ligase is used to connect between strands of DNA, and the vectors, of course, is the carrier of foreign genes or the alien genes carrying desired traits or characters. And of course, the host is you're able to able to insert the recombinant DNA so that it will replicate within the host. So these are the different technologies in the recombinant DNA. All right, so let me proceed now with the cloning vectors. All right, so what is a cloning vector? So let me enlarge this presentation. So there you have it. And let us have my pen now. Enter, use my pen. All right, so what is a cloning vector? A cloning vector is a small piece of DNA, a piece of DNA taken from a virus. It could be from a virus or a plasmid of a bacteria that can be stably maintained in an organism and into which a foreign DNA fragments can be inserted for cloning purposes. Actually, the common vectors are plasmids and bacteriophage. When you say bacteriophage, we're talking about the virus that infect or aids bacteria. So therefore, a cloning vector actually is a plasmid. No, it's a carrier of foreign genes. Actually, a plasmid is considered a cloning vector, and as well as the viral DNA. The virus or the viral DNA can be considered as a cloning vector that carries the genes of desired interest from the foreign gene or alien gene. So what are the salient features or important features of a cloning vectors? Okay, a cloning vectors has ability to replicate within the bacterial cell independent of a chromosomal DNA. Ability to replicate within a bacterial cell. Para mapadaghan natin siya, we cannot clone the recombinant DNA or the foreign genes or the foreign source of gene if it is not replicated in the bacterial cell or the host cell. Okay, so another features of the cloning vectors, high copy numbers per cell. So, dapat marami tayong vectors doon sa cell. Maraming plasmid na makakuha natin. For example, in a bacteria, dapat meron tayong ma-obtain na plasmid doon in a certain cell of bacteria. As in fact, sabi dito, plasmid should be 15 to 100 copies per cell. Para daghan tayong ma-harvest na plasmid, if we could not harvest any plasmid from a bacteria, and there's no way that we can produce a recombinant DNA, or we cannot clone the new genes into the host. Okay, so another salient features of a cloning vectors: alien DNA, when integrated with vector, can multiply equal to the copy of the vector. Meaning to say, the vector is a plasmid. When a foreign genes inserted into a plasmid, and when the plasmids containing a foreign genes, it becomes now a recombinant DNA. And when that recombinant DNA is inserted into the host cell, it will replicate at the rate of the replications of the host cell. But ang na-replicate doon actually yung plasmid that carrying the foreign genes. So therefore, the alien DNA, when integrated with the vector, can multiply equal to the copy number of a vector. When these numbers of vectors replicated also has equal numbers of that genes replicated, because it is the vector that carries the genes of the foreign genes. Okay, so another salient features of the cloning vectors: vectors can be engineered for easy linking for DNA, for foreign DNA, and selections of recombinants from non-recombinant DNA. So we can engineer the vectors, no, for easy linking of foreign DNA. So, for example, the plasmid, it is a ring structure. We can cut a certain portions of the plasmid. We have to re-engineer for that. So we remove something, and we have to replace that with something. No? That is a re-engineering of the plasmid or the vectors, such that it facilitates the insertions of foreign DNA and selections, and later on we can choose which of these following plasmid exhibit these particular traits associated with the genes inserted into it. Alright, so let's proceed. So this is how the gene cloning with different vectors. In this particular illustrations, we used vectors, bacteria, and as well as the virus. No, so in the bacteria, in the bacterium, we remove the plasmid. Then plasmid becomes our vector, and we have to cut our portions of our plasmid using the restriction enzymes. No, we have the endonuclease and the exonuclease. Then, and the cutted portion, we have, of course, we have to cut the portion of the DNA of the source. So we have to obtain the gene from the DNA of the source. That's why I have the foreign gene inserted into the cutted portions of the plasmid, so that when they are bind together using through the use of the ligase enzyme, they become now a recombinant DNA molecule. When this DNA molecule inserted into the host cells, and when that host cells divide, and the recombinant DNA participates or joins the cell divisions in the host cells, that's why we have now the cloned, we have the multiply the recombinant DNA to the replications within the host cells. No, so it allows replications within the host cell. That's why this bacteria, or this vector carrying the traits of interest, it's now associated in this resulting cell after a cell division. Alright, this is for the virus at the bacteria. Then we have another gene cloning using virus as a vector. Look at this one, we have the viral DNA. Okay, then in the viral DNA, we have to cut a specific portion, of course, the same. We have to use the restriction enzymes. Okay, we have to cut a certain portion. We have to re-engineer that. And of course, another portion from the source of desired gene, from a foreign gene, we have to cut using the restrictive enzyme. Then we have to join them using a ligase enzyme. Then we have now recombinant DNA molecule, recombinant viral DNA molecule. Then that viral DNA molecule will be introduced into the host cell, packaged into a mature virus by transfixion. So we have to do some infections of the host cells so that infected cells with the viral DNA, it undergo lysis or bursting or decomposition. Then it will burst, resulting into numbers of recombinant DNA molecules containing the viral DNA molecule. This one usually happens in animals, no? It usually happens in animals. So, such as for example, in animals, such as for example in cancerous cells, no? It usually happens, no? The cancer development of cancer cells is using this mechanism. But in plants, usually ito yung mechanism using bacteria as the vector. Alright, let's proceed. So there's some features to facilitate cloning into vectors. So features to facilitate cloning into vectors. Alright, cloning happens only when features required to facilitate cloning into vectors. So dapat there must be an origin of replication, because if there is no points of replication, then there is no way we can reproduce the recombinant DNA, recombinant cells into the host cells if there's no points of replication. Because yung mga foreign gene natin na dinala ng mga plasmid at saka viruses can only be reproduced when it allows to replicate within the host cells. Okay, there must be an origin of replication, and there must be a selective marker. Why? Because when we use a selective marker in a certain portion of a DNA, so there must be a certain genes. For example, this one, no? If this is the strand, single strand of DNA, or shall we say double strand of DNA, containing different nucleotides such as adenine, cytosine, thymine, and what else, no? So for example, this is the sugar bases. So there is an, shall we say, adenine in this side, and we have the thymine in this side, we have the cytosine, and we have the guanine on this side, so on and so forth, no? So, okay, the purine should always be paired with the pyrimidine. Okay, purine bases and the pyrimidine bases. All right. So there must be a selectable marker. For example, in this particular DNA strand, a certain portion of these, there are some genes in the certain portion of these, no? There are some genes that carrying different traits, different phenotypic, expressing different phenotypic characteristics. Now, for example, in a selected marker, for example, this portions, no, carrying the gene of shall we say penicillin resistant gene, no? Penicillin, resistant gene on that particular. So in these particular portions, this is responsible for our penicillin resistant gene. Now, if this particular gene is of our interest and we try to capitalize on these genes, we try to use these genes, we have to cut the certain portions of this source of genes. We have to cut this one, so we have to select a marker. So this portion has to be cut because this portion is carrying the particular genes. There are many genes in this strands, no, structure, but we have to locate, we have to identify which specific gene is within our interest. Yun ang kukunin natin, no? So maraming mga genes dyan. So kunin natin, so selected marker para alam natin ito yung mga genes na carrying the traits under our interest. For example, ito yung genes nakunin natin, for example, mga matatangkad na mga genes na responsible for height, kunin natin ito at i-hybrid natin doon sa other source as a host cell, so that the resulting host cells will exhibit the same characteristics, having a tall phenotypic physical characteristic, for example. So there must be a selectable marker, and there must be a cloning site, because a cloning site, yung for example doon sa plasmid, so i-cut natin, so mayroong cloning site. Saan ba siya natin isumpay? Saan pa so ibutang ang kanaang mga genes para mudaghan o para ma-exhibit siya nga karakter dito sa source or sa host. Okay? So another features to facilitate cloning into vectors is vectors for cloning genes in plants and animals. So there are vector for cloning genes in plants and animals. Actually, we have the bacteria, and of course, they have the virus as vectors. So that we'll try to look at it individually. Ito yung sinabi ko kanina. So origin of replication, sequence of DNA where replicate starts and initiates. No? So in a particular, for example, in this particular gene, no? So there must be a sequence in here that asa ba dapat mag-initiate. So i-cut natin ito, then insert natin dyan ang ating parent cells, at dito mag-initiate ang asa ba nga portion mag-initiate ang replication. Okay? Then alien DNA when linked to it, when alien DNA when linked to the replication site, it replicates with host cells, you know. Then the origin of replication regulates and control copy number of linked DNA or alien DNA. Okay. So another, we have the selectable marker. So a selectable marker identify and eliminate non-transformants from transformants. Pag sinabi natin non-transformant, ito yung genes na normal genes na hindi natin siya minodify. Pag sinabing transformant genes, ito yung mga genes na of interest na modify natin, change natin, na-alter natin. And directly speaking, this is the recombinant DNA, no? Itong mga transformants. And selectively permit growth of transformants. Okay? Alright, so transformations, this is a procedure through which the recombinant DNA introduced into the host bacterium. Again, transformation, the procedure through which the recombinant DNA introduced into the host bacterium. Then, until such time, yung host bacterium receiving that recombinant RNA, it will now express the phenotypic expressions of the genes of interest. Okay? When we say phenotype, we're talking about the physical expression of the genes under or within our interest. For example, in a certain strand of DNA, there's a gene for a tallest gene, tall gene, yung mga matatangkad na mga tao. So, ini-extract natin yung genes responsible for taller height. Tapos, i-inject natin doon sa gene ng mga maliliit na tao. And eventually, yung cloning na yun, mag-clone yung tao na yun, yung maliliit na tao, meron siyang physical appearance na matangkad, which is the expression of the genes, the tallest genes na doon sa genotype. Again, meron tayong genotypes is the genes, and ang phenotype, ang physical expressions of the genes. Okay? An example in here, we have the Escherichia coli. So, there's an Escherichia coli or E. coli resistance to ampicillin. So, pag resistant siya sa ampicillin, dili siya mamatay sa ampicillin. And also, naapoy resistance sa chloramphenicol. Kana nga E. coli, dili siya mamatay sa chloramphenicol, kaya resistant man siya. Then, you also have the resistance to tetracycline and kanamycin. No? So, selectable markers which is lacked in normal E. coli. So, meaning to say, yung itong mga engineered or recombinant E. coli, doon sa structure ng kanyang DNA, merong mga genes doon na responsible for resistance against ampicillin, chloramphenicol, tetracycline, kanamycin, no? Uh, Pag kukunin natin yun, so under that particular DNA of that organism, pag kukunin mo yung genes responsible for ampicillin, pag kinuha mo na, yung organism na yun, mamamatay na siya kung mayroong ampicillin kasi kinuha mo na yung traits responsible for resistance of that organism against ampicillin. Okay, yun po ang concept doon, no, sa resistant. And also, pwede natin kunin yung genes within the DNA of that E. coli na resistant na ampicillin, i-transfer natin sa ibang organism so that yung host organism na pinaglipatan natin ng genes ng resistance against ampicillin, so it will now exhibit the traits or the characteristics the same to the E. coli that has now resistance to ampicillin. So, ganun lang yun. Okay? So, cloning sites as one of the features that to facilitate cloning vectors. So, cloning site, this is the region in the vector. So, it is the region in the plasmid or in the viral particles or the viral DNA. It is the region in the vector where ligation of target DNA takes place. So, cloning sites, or this is the insertion site. No, doon natin i-insert ang ating foreign insertion site doon sa DNA strand. So, insert natin doon para pag mag-replicate yung DNA na yon, so ma-replicate niya kasama yung traits na in-insert doon sa DNA niya. So, magiging genetic constitution na niya, magiging genetic material na ng DNA na yon, so that that particular cell carrying that DNA na mayroong foreign gene, so i-express na niya ngayon yung characteristic ng foreign gene. Okay? And of course, ang cloning sites created commonly by specific restriction enzymes at the recognition site or restriction sites within a vector where ligations of alien DNA takes place. Kasi ganyan eh. Pag mayroong portion doon sa plasmid, i-cut mo yung portion ng plasmid, portion ng DNA niya, open na ngayon yon. So, ilisan natin ng bagong genes from a different cell. So, yun, pag mag-replicate na, maklone na yung genes na yon. So, we have some different examples in here, no? That we have the PBR 322. This is a genetically engineered plasmid, no? Genetically engineered plasmid, sinabi kanina, we can hybridize the plasmid, no? Genetically engineered plasmid, widely used E. coli cloning vectors. So, E. coli, ito yung mga E. coli, actually is Escherichia coli. Actually, these are the bacteria from the feces from our stool sa atong mga dumi, no? Ito yun, sa mga dumi ng hayop, dumi ng mga animals. Okay? So, yes, widely used is the Escherichia coli cloning vectors. That is the ampicillin-resistant gene encodes for as ampicillin resistance. And you also have the TR or the tetracycline resistant, no? And have specific restriction sites for different restriction. So, yung mga plasmid na yon, yung engineered plasmid na yon, ito yung plasmid natin. So, itong mga plasmid na to have specific restriction site for different restriction endonuclease. Merong mga site dito for terramycin resistant, tetracycline resistant. So, nang support, merong mga region sa plasmid na mayroon siyang characteristic carrying the gene of with different characteristics. Okay? So, for example, ito yung mga site, no? Yung mga site, restriction site na pwede natin i-cut, pwede natin i-cut at i-insert doon sa iba, o pwede natin alisin, no? Para, yun. So, presence of more than one recognition site in vector will complicate the gene cloning. Now, kung maraming site doon na mayroon siyang recognition site, so mahirap yun kasi mag-complicate ng gene cloning natin. But kaya naman yun, no? So, ligations of alien DNA. So, paano ba yung alien DNA i-insert, i-join, i-connect doon sa ating plasmid, no? So, of course, ligations of alien DNA, restriction sites present in one of two antibiotic resistant genes. No? In particular, doon sa antibiotic resistance, no? Focus kasi tayo sa example natin sa antibiotic resistance at as resistant to ampicillin, chloramphenicol. No? Okay. There is a restriction site present in one of two antibiotic resistant genes. So, may mga site doon responsible. For example, this is the DNA and that portion of the DNA merong mga genes na resistance against antibiotic. Yung mga specific site ng DNA na mayroong genes doon, so meron siyang kanya-kanyang characteristic, no? So we can inactivate the gene, no? We can inactivate the gene. For example, if we are to remove that gene from the DNA, therefore, inactivate na ang gene na ngayon kasi hindi na siya ma-express doon sa kanyang mga resulting offspring, no? Hindi siya ma-express kasi tinatanggal na yung mga genes na yon. So, pwede pala, no? Pag mayroon we are humans na mayroon tayong sa kaliwatan natin sa ating mga ancestors, mayroon tayong pangit na mga characteristics, shall we say, mayroon tayong kaliwat na shall we say kanang mayroong bad traits, no? So, yung bad traits na yon nandiyan sa family, so naka-embed yan doon sa DNA natin as a specific gene. So, pwede natin kunin yung gene na yon para ang succeeding generation natin hindi na i-exhibit, hindi na mag-manifest yung bad traits natin kasi na-remove na natin doon sa portion ng ating DNA as a bad genes, no? Inactivate, no? So, inactivate the gene, enable to choose transformants from non-transformants. For example, ang isang organism, resistant siya sa chloramphenicol, so sa ampicillin, pag niremove natin yung gene na yan, na resistance against sa ampicillin, dyan sa DNA structure na as a gene, so yung resulting na mga anak niya, mamamatay na ngayon yan na sa ampicillin kasi hindi na sila resistant. Yun. So to inactivate the gene, enables to choose transformant from non-transformant. So, pwede lang siya i-activate, pwede rin i-activate. In this particular example, so this is example of plasmid, no? With different sites, with recognition sites, no? With different sites. Maraming sites dito. So, we can remove this one, this particular site, the BAM each one and the SAL each one. These particular portions of the vectors, particular portions of a DNA vectors, this is the genes responsible for tetracycline resistant. So, ibig sabihin, if this is an organism, itong organism na ito, hindi siya mamamatay ng, it cannot be controlled, it cannot be killed by a tetracycline antibiotic because mayroon siyang resistant gene. So, pwede natin tanggalin natin ito using the restricted enzyme. Pag tinatanggal natin ito, mawawala na ngayong tetracycline resistant. Now, pag wala na ito, yung mga succeeding offspring niya, mamamatay na yan ngayon when added with a tetracycline. Ganun. Pwede rin ito kunin at i-insert natin sa other cell, sa other DNA para yung resulting DNA kung saan natin i-insert itong portion na to will carries the traits that is tetracycline resistant. Ganun din ang iba dito, no? So, yun po ang ibig sabihin doon. Hindi ko na lang siya clearly discuss. For example, ito, E. coli, itong portion na to, It has a different traits. Ito naman, merong traits. Merong traits. Ano bang ibig sabihin nito? We can remove this one. We can inactivate. We can deactivate this one. Okay? So, yun, Then, vectors for cloning genes in plants and animals. Ano yung mga vectors na gagamitin natin to clone genes in plants and animal? Actually, no, we have been using viruses and bacteria. Okay, virus and bacteria transform eukaryotic cell. Okay, eukaryotic cells by delivering genes into the host cells. It be plant cells or animal cells. When we say eukaryotic cells, we are talking about an organisms in which the organelles within the cell is bound by a membrane. Again, when we say eukaryotic cells, we are talking about the cells by which the organelles is bounded by a membrane. No? Example of eukaryotic cells, we have the fungi, we have the protozoans, we have the plants and animals. Tayo po mga tao, ang cells natin are mostly eukaryotic cells. No? Para hindi diffuse yung mga boundary ng ating organelles. Okay? Well, compared to prokaryotic, usually yung mga prokaryotic, yun yung mga bacteria, no? Yung mga organelles niya wala siyang distinct membrane, no? Katulad ng, for example, ang nucleus ng prokaryotic, walang mga membrane, parang diffuse. But ang eukaryotic, ang nucleus niya is merong mga nuclear membrane na klarong-klaro, no? So, bacteria and virus transforms eukaryotic cells by delivering genes. No? Usually, ang ginamit kasi na plasmid is bacteria and virus. An example in here is the Agrobacterium tumefaciens. This is the crown galls or the pathogens of a dicot plants. No? The Agrobacterium tumefaciens deliver a DNA called tDNA and transform the normal cell into a tumor which produces chemicals required by the pathogen. Mean to say, ito palang Agrobacterium tumefaciens, this is a bacteria responsible for the for the formations of tumor. So in a plant, no? Sa plant. Kasi gusto ng bacteria magka-tumor kasi gusto nila yung tumor produce chemicals na magagamit nila at their advantage, no? Excuse me. So in the plants, so we use the bacteria. No, an example of that is the Agrobacterium tumefaciens, so that it will delivers genes into the host cells, resulting for the modifications or the alterations of the genes of the plants, no, of the host plants. But in animals, naman, it is the virus that transforms the DNA of the host animals, no? Using the retrovirus. So retrovirus transforms normal cell into a cancerous cell. Pag nag-cancer pala tayo, that's because there is a retrovirus in our system that transforms normal cell into a cancerous cell. So retrovirus is for animal, bacteria are for plants. Alright. So an understanding of mechanisms of delivering genes by pathogens to their host can help to understand the tools of pathogen and can be used to deliver genes of interest in human. So in a tumor-inducing plasmid, such as in the Agrobacterium, no, the tumor-inducing plasmid or the T1 of Agrobacterium tumefaciens modified into cloning vector. Again, itong Agrobacterium tumefaciens modified into a cloning vector, itong plasmid na to. Non-pathogenic to plants and have ability to deliver genes of interest to variety of plants. So, pwede siya magamit for cloning, for recombinant DNA, ang Agrobacterium tumefaciens. And retrovirus, on the other hand, sa animals, it will disarm, retrovirus disarm the immune system of the animals, then delivers the genes of interest in animals. Ayun, no? Retrovirus disarm the host cell and delivers the genes of interest in the animals so that the resulting animals, ang genes niya, when replicate, nagiging cancerous cells na, no? So, when gene of interest is ligated into a suitable vector, so transferred into a suitable host, it will multiply. Now, when the genes of interest is connected or ligated into the vector, then transferred into the host, then when the host multiply, then also the genes of interest also participate in replications and also multiply. Okay? So ito yung plant vector using the T plasmid from the bacteria. Ito yun, the T plasmid. Cut siya using the restriction enzyme. Inserted natin ng new genes. Then inserted in, magiging recombinant DNA na ito. Then inserted into the host cell. Okay? Then when the host cell divide, meron tayong plant with new traits, o pwede tawagin ito na transgenic plants ngayon. So, transgenic. Kasi, na-modify na ngayon yung DNA niya. So, parang meron ng nasagol doon. No? One of the examples of the transgenic crops that we have, particularly in the Philippines, we have sa mais, no? We have the BT corn. BT corn. Example of that, we have the Bacillus thuringiensis corn. Itong corn na ito has been designed so that kung ang ulod mukaon sa mais, ang ulod mamatay. So, di siya kadaghan because transgenic na siya, mayroon na siyang organism nilagay na makapatay siya sa ulod sa mais. No? Ang ulod, gitawag ng ulod sa moog, Ostrinia fornacalis. But just never mind on that. Marami na tayong transgenic crops, no? Marami tayong transgenic soybean para mag-produce ng feeds. Meron tayong transgenic canola para mag-produce ng oil. So, meron tayong mga ganyan na, no? In our present generation. Yan, at least ito yun sa tanom. But doon naman sa animals, of course, we use the retrovirus. This is the retrovirus or the infected virus na ito, no? Retrovirus, nag-invade siya sa cell. Invade siya. Okay, pag invade niya sa cells, ang host cells, itong cells na to containing the viral particles, the viral DNA. Then, yung viral DNA, yung DNA niya, yung strand niya, reverse transcription, then multiply. Dayon, meron tayong viral RNA, meron tayong RNA-DNA hybrid, hanggang sa yung insert siya nucleus and mag-ligase, mag-join doon sa strand ng DNA ng cell inside sa nucleus until such time na ma-infected yung cells na yon at pumutok, burst or lysis. Then, yung pagputok niya, marami ngayon niyang ma-produce na viral cells. Ayun po ang mechanism ng animal vector, particularly for the mechanisms of cancerous or viral cells multiplications within the host cell. Ayun po. Okay? But of course, it requires a competent host for transformations with the recombinant DNA. Now, pag yung recombinant DNA na yon, from the plasmid, pag hindi siya maka-penetrate, hindi siya maka-insert doon sa host cell, so wala rin mangyari. So, kailangan mo na niya maka-penetrate doon. No? So, as a competent host, the recombinant DNA should enter cell to transform cell. Kung di siya maka-insert, di siya maka-transform. No? So, there are many points of entry. No? For example, sa ating ngayong COVID-19, so it can enter in our nose, in our eyes, in our mouth. So, that's why kung maka-enter siya sa ato ang cell, ayun, mag-multiply, kaya nga mahirapan na tayo, no? But DNA cannot enter the cell in a medium without treatments. So there must be a treatment, there must be some requirements so that the recombinant DNA can penetrate into the host cell, no? The host should be made competent to take up DNA. So paano magiging competent kayo yung host cells para mag-take up siya ng DNA? So ang gagawin lang is that so there are different techniques to make host competent. Use the chemical treatment, no? Uh, dito, no? Treating host bacteria with specific concentrations of the valent calcium, lagyan ng calcium to increase efficiency and make DNA enter to the cell wall of host, no? Pwede rin na heat shock, no? Heat shock, sudden change in the temperature. So, through heat shock, mixtures of recombinant DNA and host cells. Pag mag-mix na ngayon yung recombinant DNA and host cells, so meron na ngayon yung transformation, no? Meron na ngayon yung association between the host cells and recombinant DNA. And also, sa chemical treatment, no, it requires incubation of host and the RDNA, parang i-homol sila, parang isagol sila, no, idugay on an ice and then placing them at certain temperature. Then, enable bacteria to take up our recombinant DNA. And because of the chemical treatment, so mapadali yung entry ng bacteria, ng take up ng bacteria ng recombinant DNA. So another methods to make the host competent is for the microinjection, no? So inject natin yung recombinant DNA directly injected into the nucleus of the host cells. Parang i-inject siya. Microinjection. And this is common for animal cell, no? And you also have the biolistics and gene gun. So the host cells bombarded with high velocity microparticles of gold or tungsten para po sila ng tanom, no? So microparticles are coated with itong mga particles ng gold and tungsten actually coated with recombinant DNA para makasulod siya doon sa host animal cell, no? Used for plant cell. Another techniques used to make the host competent so that it will take up the recombinant DNA is the disarming pathogen. So try to do that. So this is the technique to introduce recombinant DNA, microinjection, no, directly to the nucleus. And biolistic, no, in plants in particular. Okay. So that's it, no? So, and we have in here the process of recombinant DNA technology, no? In the process of recombinant DNA technology involves following steps. Of course, we have to isolate the genes of interest from the source. Fragment natin yung DNA, i-cut natin into a portion using the restricted enzyme, restriction endonuclease. Pag nag-cut na, i-isolate natin using the electrophoresis, so isolation of desired DNA fragments, because naidagko, naigagmay. And we have different fractions or fragments of the genes carrying different characteristics, so you have to select a particular gene. So pag separate, you have to select. Then, pag select na natin yung particular genes, ligation of DNA fragments into a vector. Insert natin sa vector yung mga nakuha nating genes na kailangan natin i-express sa host. Then, transferring the recombinant DNA into the host. Then, culturing the host cells. Ipadaghan na yun siya. At the medium-large scale, the extractions of desired product. Ipadaghan natin, no? So, yung kanina, no? So, pag naana siya dito, cutting of the DNA in the location, yung mayroon tayong mga select markers, no? So, ito yun, no? I-digest, coag plasmid, then plasmid, siak, kaya atong insertan siya og new genes, then i-process through electrophoresis. Because the DNA is negatively charged, therefore, it will attract to a positive charged particles. In electrophoresis, at the anode side, we have the anode and the cathode. At the anode side, or the positive charge side, doon mag-attract yung mga DNA natin. Okay? Then ligations of genes of interest and DNA vectors. Isumpay ang foreign gene and DNA vector inserted into the host so that it will multiply. It will be produced in a large batch, no, in a large amount. Then we can amplify also the genes of interest using the PCR, the polymerase chain reaction. So mapadaghan natin, amplify into 1,000 times, million times, billion times. Meron tayong isang gene. Okay, for example, the PCR, the polymerase chain reaction, technology used to amplify a single or new copies of DNA to generate thousands to million of copies of particular DNA sequence in vitro in laboratory. In our times today, we have COVID-19. No? Kuha tayo ng swab, kuha tayo ng sample sa ating ilong o saan ba makuha ng sample. Na yung sample na yon, pinaprocess, we have to obtain the DNA, the viral DNA. Then we have to replicate that viral DNA para mastadihan natin, malaman natin kung ano na ang state of the viral infection, no? Kung mayroon ba o wala. And of course, sa amplifications of gene of interest, kailangan natin ng components required. We have primers. So itong primers na to, para mo-complement siya, no, sa strand. Example, sa primers, we have a single strand. Meron tayong nucleotides na A, single strand. Meron tayong T. Meron tayong guanine. Meron tayong cytosine. Meron tayong guanyl. These are the nucleotides in the phosphate, sa sugar phosphate bonds of the DNA. Now, kung mayroon tayong single strand, it always partnered with, for example, kung mayroon kayong adenine strand, it is always partnered with the pyrimidine strand. Adenine and pyrimidine strand, these are the strand, no, of DNA containing different nucleotides. And nucleotides that we have adenine, thymine, cytosine, and guanine. For example, this is the single strand DNA, and because of the primers, itong primers na to, it will identify, it will complement the other side of the strand. For example, lagyan natin ng primer, ito walang partner na strand ito. Pag lagay mo ng primer, automatic ito maglagay ng T is thymine, kasi thymine ang always partner ng A sa DNA is thymine. Dito naman sa thymine sa kabila magiging adenine. Ito namang cytosine magiging guanine. Ito namang guanine magiging cytosine. So ito yung ngayong resulting new strands that has been formed when we add primers into that particular single strand DNA. Okay? A component required. Now, primers, small chemically synthesized oligonucleotides that are complementary to the regions of DNA. So, complementary, ibig sabihin mo, complete siya to that region. Katulad sinabi kanina, single strand lang ito. To complete the double strand, so lagyan din ng primer para makomplementary, makomplete niya ang region ng DNA. So, nucleotides and DNA polymerase incubated together. So, parang sagol sila doon sa nucleotides o sa single strand ng DNA para mahimo siyang double strand DNA with the use of the primers. Now, the steps in PCR, no? The steps in PCR, we have denaturation, no? So, ibig sabihin ito, denaturation of a double-stranded DNA by heating. So, merong double-stranded DNA, by i-heat siya, ma-denatured siya, then mahiwalay ngayon yung individual strand sa double-stranded DNA. So, mabulag yung mga strand, no? Na strand ng purines, ang strand ng pyrimidine, mabulag sila. Okay? Until after nabulag, so nahiwa-hiwalay niya, single strand na siya, lagyan siya ngayon ng primer para makomplete yung partner, the other side ng strand ng DNA, and that process known as annealing, no? Then we have to add a DNA polymerase. Anong purpose ng DNA polymerase sa PCR? Polymerizations with nucleotides in a medium genomic DNA acts as template. For example, itong single strand as a template, pag i-polymerize nimo, so yung mga naka-single strand DNA, using the DNA polymerase. So, makomplete yung kulang or masumpayan, no, para ma-extend yung strand ng DNA. Paglagay mo ng primer, makompleto yung kulang na partner. Pag nilagyan mo na ng DNA polymerase, so ma-extend yung strand na yon. So, mataas ang strand ng DNA. Okay? Then, of course, replication, repeat many times. We can multiply the DNA many times, billion times, thousand times. No, we can amplify that. So this particular example is the PCR. So we have, for example, this particular region to be amplified natin itong region na ito, ipadami natin ito. So ang gawin natin, we have to denature through heating. Pag na-denatured, nabulag siya. Okay, into single strand, nabulag. Then a certain portion of that is lagyan natin ng primers para ma-complete itong mga portion nito. If this is an A, dito na sa kabila is T. Adenine, thymine, guanine, cytosine, no? Cytosine. So ma-complete siya with primers ang kanyang complementary strand. If this is the purine strand, sa kabila mayroong pyrimidine strand, no? Ma-complete. That is an annealing process. Pag gusto natin ma-extend ito matapos ang pag-fill up ng other strand of the DNA, so lagyan natin ng DNA polymerase para ma-extend. Katulad ito, maliit lang. So na-extend, nataas siya. That is an extension with the use of DNA polymerase. Ayun, nadaghan na siya. Imbis isa lang siya ka-cell dati, isa ka double strand. Ngayon, duha na sila ka double strand. I-repeat ang cycle to amplify into a billion times more. No? Ipadaghan natin siya for batch preparation of that particular biotechnological particles, for example. Okay? Then of course, we can insert the recombinant DNA into the host cells. Insert natin siya dun sa bacteria, insert natin sa plant cells and animal cells. So, katulad nito, for example, we have transform cell. We have this one is the ampicillin resistant, and this one is not an ampicillin resistant. Pag lagyan silang agar, no? This is the growth medium, agar, and ilagay natin growth overnight. And because itong isa is resistance to ampicillin, so siya ang mabuhi. But this one, itong mga non-transformed cell, di siya mabuhi because they are not resistant to ampicillin. So now, ang nabuhi is the ampicillin-resistant colonies. Ito yung sinabi kanina that we can separate the transformed from the non-transformed organism. Obtaining the foreign gene product. Paano tayo mag-obtain? Of course, we have to use so obtaining the foreign gene product from sa ating host cells, no? The alien DNA binds with cloning vectors at T plasmid. The recombinant DNA transferred to the suitable host. So, merong host. Then the resulting recombinant DNA expresses itself under appropriate optimal conditions and after cloning of the recombinant DNA to a particular number of copies. Then target protein produce large scales of protein obtaining the alien or the recombinant DNA. Okay? So yun, ipa-produce natin ito marami, kaya mayon tayo gamit ng mga bioreactors para panabihin ngayon yun yung mga particles na yun. And these particular examples of the bioreactors, no? That one. Then another, the last process is downstream process, no? So downstream refers to the recovery and purifications of biosynthetic products, particularly pharmaceuticals. So the downstream process, the recovery and purifications of biosynthetically produced product. For example, katulad na ng mga okay ka ba tiyan, yung lactobacillus. So merong downstreaming process yon. Okay? In this particular example, ito yung mga example ng bacterium and how it is being cloned using the RNA. Nevertheless, this is all about the biotechnology discussion, and I hope you understand the discussion. And well, try to view all the discussions, and also try to look at our presentations and do some additional readings online for you to augment or for you to increase your understanding in biotechnology. And by the way, class, I will require you to produce biotechnological products out from the living organisms. For example, I want you to produce the what is this? The virgin coconut oil, the BCO. I want you to produce that, and any other products. But pwede na, BCO, pwede na. You can produce BCO at 1 liter. That would be fine for me. Submit nyo lang. Lagyan nyo ng procedure. But I will give you another instruction for that. But for this discussion, shall we stop the discussions, and I will make some announcement later on. So thank you for listening and have a nice day.